Entomopathogenic fungi form the largest group of insect pathogens, playing a vital role in regulating insect populations in ecosystems. Fungal pesticides, particularly those belonging to the genera Boveria and Metarhizium, have been developed as eco-friendly and sustainable biocontrol agents against agricultural insect pests and vectors of human diseases. Unlike other entomopathogens such as bacteria, viruses, protists, and microsporidia, which infect their insect hosts through the midgut, entomopathogenic fungi are traditionally thought to invade insects by penetrating the integument in the classic theory. Although the integument infection has great advantages in the biocontrol of pests with piercing and sucking mouthparts, the effectiveness of topically applied fungal insecticides is influenced by various adverse environmental factors, including low humidity, extreme ambient temperature, and solar UV light. Therefore, the pursuit of alternative routes for fungal infection is of great significance to improve the efficiency of fungal insecticides. The insect gut is moist and relatively stable, which can facilitate fungal conidial germination and growth. Despite efforts since the 1930s to identify fungal strains capable of infecting insects through the intestinal tract, convincing evidence with explicit histological information to support fungal gut infection has been lacking. It is generally believed that the unsuitable pH environment, numerous digestive enzymes, and diverse intestinal microorganisms in the insect intestine may inhibit fungal conidial germination. To investigate whether gut bacteria act as a protective barrier against invasion by B. bachiana in the larval gut of a Lepidopteran model insect, silkworm B. mori, we began our investigation by selecting a highly virulent strain of B. bachiana BBM1 against silkworm larvae through insect bioassays. 5th instar day, 2 silkworm larvae were fed fresh mulberry leaves inoculated with fungal conidia, and then reared under constant low ambient humidity conditions. Surprisingly, per-OS administration of BBM1 resulted in infections in both silkworm larvae without gut microbiota and untreated larvae with gut microbiota at comparable rates, indicating that the presence of the gut microflora had no obvious impact on per-OS infection by BBM1. To validate the intestinal infection of silkworm larvae by BBM1, we orally inoculated fungal conidia using a 2μl micropipetter. Only BBM1 and BBM2 could infect the larvae through per-OS inoculation. The intestinal infection not only killed silkworm larvae and Clanus bilineata larvae rapidly, but could also kill older instar larvae and pupae. Importantly, simultaneous infection via both modes of inoculation significantly enhanced the killing speed against the larvae compared to either topical or per-OS inoculation alone. The insect intestine is divided into foregut, midgut, and hindgut. Typically the foregut is subdivided into a pharynx, an esophagus, and a crop. Although a few conidia were observed in the midguts and hindguts of per-OS inoculated larvae, no hyphal growth and penetration of the midgut and hindgut wall was observed, indicating that the midgut and hindgut is not the invasion site of BBM1. Unexpectedly, numerous penetrating fungal hyphae were found in the foregut. These results indicate that the foregut is the sole intestinal invasion site for BBM1. Fungal conidiophore morphology and phylogenetic analysis of the ITS1, 5.8s, ITS2 rDNA sequence reveals a close relationship between the enteropathogenic strains BBM1 and BBM2 and non-enteropathogenic strains of B. bachiana. However, the conidial sizes of BBM1 and BBM2 are significantly larger than those of non-enteropathogenic strains. Based on these results, we designated BBM1 and BBM2 as a variety of B. bachiana, specifically denoted as B. bachiana var. Majus. Conidial germination is the initial step for fungal pathogenesis. The gut environment in most Lepidopteran larvae is highly alkaline, typically ranging from pH 10.0 to 11.5, creating an inhospitable setting for pathogen colonization. We found that BBM conidia exhibit rapid germination under alkaline conditions. Generally, fungal alkaline tolerance is regulated by pH responsive pathway. Comparative genomics analysis showed that a 249 BP fragment deletion was identified in the downstream adjacent gene of BBM pal-C. This downstream adjacent gene with a 249 BP deletion was significantly upregulated in alkaline conditions. To investigate the effect of BBMIA on BBM-PAL-C transcription, we deleted BBMIA in the BBM1 genome. Loss of BBMIA inhibited the upregulation of BBM-PAL-C after alkaline induction compared to that in BBM1-WT, indicating that the induction of BBM-PAL-C under alkaline conditions relies on BBMIA. In contrast, the expression of BBMIA replaced in RCEF252 was significantly higher than the native BBIA. These results indicated that the absence of the 249 BP fragment enables BBMIA to function as an alkaline inductive activator that significantly boosts the expressions of PAL-C. PAL-C is required for the PACC activation at the proteolytic processing level. Consistent with the proteolytic cleavages and protein localization of PACC, the expressions of PACC-binding genes involved in tolerance to alkaline pH, such as DFG5, GRG1, SIDA, and ZRT1 were highly induced under alkaline condition in BBM1. The insect foregut is discovered as a previously unrecognized route for fungal invasion. Boviria bashiana var. Magus can infect Lepidopteran larvae through both the integument and gut. BBM has evolved alkaline tolerance via a novel pH-responsive IAPAL-C-PAC-C cascade to colonize the hostile foregut. This breakthrough novel finding not only subverts the traditional theory and understanding that entomopathogenic fungi can only infect insects through the integument, but also lays the foundation for the development of a new generation of fungal insecticides and for designing novel strategies for the management of destructive insect pests. Dr. Yiling Lai, PhD candidate Weilu Zheng and Yitong Zheng from CAS Center for Excellence in Molecular Plant Sciences, and Haiquan Liu from Jiangsu University of Science and Technology are the co-first authors. Professor Sibao Wang is the corresponding author. Dr. Shuang Chu, Dr. Lily Wang from CEMPS, and Professor Muwang Li from Jiangsu University of Science and Technology participated in the research. This work was funded by the National Natural Science Foundation of China, the National Key Ran-D Program of China, the New Cornerstone Science Foundation, the Chinese Academy of Science, and Youth Innovation Promotion Association CAS. We thank Professor Raymond Street Ledger and Dr. Brian Lovett from University of Maryland, Professor Marcelo Jacobs Lorena from the Johns Hopkins University School of Public Health for their comments and suggestions on the manuscript. We also thank Professor Bo Huang and Professor Zhengji Li at Anhui Agricultural University for kindly providing some fungal strains. Welcome to download and read our paper.